Welcome to Are You an Accident of Nature? Part 4, No Information by Chance, DNA. This is one of my favorite talks because in this we dispel the myth that you can get information by chance. You know, it's easy to say that. It's required to believe that for evolution. That's what evolution is based on, the idea that you can get information by chance plus natural selection. And that is provably false because we've now advanced in genetics and the understanding of DNA to understand how it stores the information for proteins. And we can calculate the probabilities, and we can prove mathematically that we could not get the information for a single typical protein by chance, not in the whole history of the observable universe. We're going to get through, by the end of the talk, you will understand, Lord willing, that there's a better chance that you could pick a single atom out of the observable universe than you could get the DNA information for a single typical protein by chance. It's not remotely possible. If you can't get one, you can't get thousands or hundreds of thousands. So, no information by chance, DNA. Information is not created by chance. That's a fact of information science. It's provable. And when I say that, I mean like in meaningful amounts, like a sentence or a protein. I'm not talking about a word like by, B-Y, 2 letters. I'm talking about like a sentence or a protein. A word like by is meaningless. And if that's all you had for information, you'd never get life. And you can't do it in such a way that it adds up, because I'm talking about the totality of every atom that ever existed in the universe, in the observable universe. You could not get the right information for a single typical protein. So we know for certain the way DNA codes for proteins. We know the probability of a correct code for a protein. And we know that it's impossible that chance plus natural selection could create even a single typical protein. So information is not created by chance plus natural selection. Now, how do we know that? Well, the information of life is stored in DNA. As the field of genetics has made great strides in understanding DNA, but I think it's fair to say we still don't know what most of it does. It is fantastically complicated. We're making advances, but the information in DNA is very amazing. But we do know how it codes for proteins, so we can make some measurements and, in a sense, quantify it in some fashion. Mutations plus natural selection can produce small changes in information, but it cannot produce significant increases in complexity. Okay? You can get changes in information by mutations plus natural selection, but you cannot produce significant increases in complexity, like to change one kind of creature, say a mouse, into another kind, say a bat, something like that. That's not possible, not in billions or trillions of years even. So we know that thousands of mutations, and many of these create diseases. For example, sickle cell anemia, cystic fibrosis, color blindness. These many diseases caused by mutations, but there's no known mutations that increase information. What's the chance of spelling a typical protein correctly by accident? By spelling, we mean getting the right DNA sequence for that. And that's what we're going to look at in some depth, and I think you'll understand it, and I think this will be a revelation to many people who have not been properly or well taught, people that take university courses in genetics, in evolutionary biology, and they've never understood how DNA codes for proteins and how that makes it possible to mathematically disprove the theory of evolution. When we say disproving the theory of evolution, there can be small changes in information. And genes can be turned off and they can be turned on. They can be selected and deselected. But the point, and you can get tremendous variety of variations within a certain kind of creature, say within a certain kind of dog. You can get incredible variety. Big dogs, little dogs, Great Danes, Chihuahuas, long hair, short hair, curly hair. But the point is they all came from a common ancestor, not by adding new information, but by removing information. You will not get a Chihuahua, probably, to reproduce with a Great Dane. It's probably not going to work. And if you breed here's a better example. If you breed two tiny little dogs, two tiny little Chihuahuas or toy poodles, say toy poodles, you'll never get back to a Great Dane because the information for that large dog has been removed. So that's the point. So now we're going to explain a little bit about probability, in case you've forgotten your basic mathematics, because this is just not very difficult, and just enough math so we understand how to calculate probabilities. Suppose we have two gaming dice, and we say, what's the odds of getting double sixes? Well, if we roll one dice, there's 6 different possibilities on the first die, right? And with the other die, there's 6 different possibilities. So if we got a 1 on the first die, the second die could have a 1, a 2, a 3, a 4, a 5, or a 6. 6 different possibilities. If we got a 2 on the first die, there's 6 more possibilities. And likewise, if we got a 6 on the first die, there's 6 more possibilities. In all, there's a total of 6 combinations on the first die, 6 on the second die, and a total of 36 different possibilities. We multiply. We don't add, we multiply. Very key point. The number of possibilities on one die with the number of possibilities on the other die. And that tells us the chance is 1 in 36 of getting double sixes. Now what if we add another die? So we have 3 dice. Well, we started with 36 combinations with just 2 dice. And for each of those 36 combinations, on the third die, we could have a 1, a 2, a 3, a 4, a 5, or a 6. So the probability, then, of getting a 6 on each of 3 die is 6 times 6 times 6, which is 1 chance in 216. See how that works? It's multiplication. So suppose we wanted to, what are the odds we get a 3 on the first die, a 5 on the second die, and a 1 on the third die? Again, 1 chance in 216 that we get that exact combination in that order. Now, information is an exponential function in this sense. The more die you add, you multiply another 6 times, and it goes up fast. And that's the problem for information. We can think of it as an exponential function. Now, in information, order matters. For example, would you rather have $19 or $9,001? Also, we took the same numbers, 0019, and we just rearranged them. So the order of the information is very important. Likewise, do these letters mean anything to you? But if we arrange them, we would have the word information with the same letters. Order matters. You just can't have the right letters. You need the right letters in the right order for information. Now we're going to do a little experiment here. Suppose we had a huge hat, and in this hat were many letters of the English alphabet, A, B, C, D, O, S, J, etc. And suppose there was a near infinite number of each letter in the hat. What would be the odds of spelling the word information by chance? Well, there's 26 letters in the English alphabet. So the odds of getting the first letter, picking it out of the hat by chance, would be of getting an I. The first letter in the word information would be 1 in 26, right? 26 letters in the alphabet. it's equal number of each letter. The odds are 1 chance in 26 we get an I, the first letter, correctly. What are the odds we get the second letter correctly? N. Well, again, 1 chance in 26. What are the odds we get the third letter correctly? Well, 1 chance in 26. And we go on and we find that to get spell the word information correctly by chance, just by pulling random letters out of this hat filled with letters of the English alphabet, would be 26 times itself 11 times. That turns out to be a really big number, 3,000 some trillion. Let me give you an idea. Now, this is a word with just 11 letters in it. The chances that you'll die by a bee sting are about 1 chance in 6 million. The chances that you'll win the Powerball lottery at present are about 1 chance in 292 million. Nobody wins the Powerball lottery almost. Very rare. Tough to win that one, huh? The chances that you'll spell the word information correctly by chance is something like 3,000 trillion. It is very, very difficult. Better chance you'll win Powerball than you will spell a simple 11-letter word by chance. Now, what about the information in a protein? Well, the English alphabet has 26 letters. Proteins have sort of alphabet also. They're called amino acids. It is an alphabet of approximately 20 different letters. Proteins are the building blocks of life. And amino acids are the building blocks of proteins. What's an amino acid? They're small molecules, depending on what you want to call small, and they're linked in long chains to build proteins. Each protein is made up of a chain of amino acids. Now, there are 20 what we call standard amino acids. With very rare exceptions, these amino acids make up the proteins in all life. They're all what we call left-handed amino acids, which is a problem for life forming by chance, because in nature, there's both right-handed and left-handed forms of the same amino acid occurring in equal numbers. And if you get a right-handed amino acid in the mix, it ruins your protein. It will make the protein structure unstable because there's some bonding that goes on in the protein that requires all left-handed amino acids. And without that bonding, the protein will flop around too much. So we're going to ignore that problem. It's a serious problem for evolution. We're going to ignore that problem for now and just suppose that we can calculate, roughly speaking, probabilities with this alphabet of 20 standard amino acids. Here's another representation of the 20 amino acids of life. DNA is where information is stored for life. DNA is made up of a long chain of bases. A little bit more technically accurate, we could say nucleotides. Now, a nucleotide consists of a base, a phosphate, and sugar, or maybe a few of those, but basically has 3 components to it. But every nucleotide has a base, and there's only 4 kinds of bases. So for the purposes of our illustration and discussion, we can say, without sacrificing any accuracy in the calculations, that DNA is made up of long chains of bases. So we're just going to simplify a bit and use the word bases here. Now, base on one side of the DNA is paired with a base on the other side. So we have this spiral, this double helix, and it's like rungs on a ladder. And the DNA has matching base on each side of this double helix, like rungs on a ladder. There's a 2-step code for proteins. It's not difficult. Stick with me here, because in the end, this is going to be simpler than it seems at first. A group of bases specifies an amino acid. So you take 3 bases, and that will tell you a particular amino acid. So like 3 rungs on the ladder of DNA. You know the 3 rungs. If you know the 3 bases, you can just look it up in a table in a standard science book and say, okay, that's this amino acid. And so the group of bases specify an amino acid, and a group of amino acids specifies a protein. It tells you, identifies a particular protein. There's only 4 bases in DNA. They're abbreviated by the letters A, T, C, and G. That's fairly simple. Only 4 bases. So if you look at a string of DNA, geneticists will commonly illustrate it or in this way. They'll have just those 4 letters, the letters of the bases strung out in a big, long pattern. It could be thousands or tens of thousands or whatever unit they're looking at. Like here, A, T, T, G, C, T, A. But it's just those 4 letters. In English, we have 26 letters plus a space. To write words and sentences with. In DNA, it's just these 4 letters. The DNA comes in pairs. If there's an A on one side of the double helix, we know there's a T on the other side. If there's a C on one side, it always matches up with the base G on the other side. So let's take a look at this DNA sequence here. So if here's an A here, we know there'll be a T on the opposing side of the double helix. A C here means there'll be a G there. Now the amino acid code goes in triplets, codons it's called, but it just means 3 bases. That's all it means. For example, if you see CCA, that specifies the amino acid proline. If you see the bases ACC in DNA, that tells the machinery of the cell to build the amino acid 309. So that's the code. And so for each of the 20 amino acids, we have codes. And this is known. It's been figured out. So for proline, again, we have CCA, CCT, CCC, or CCG. Any of those combinations of bases will give you the amino acid proline. We also have a start code and a stop code. And the start code is ATG. So the machinery of the cell, when it's reading along the DNA, if it sees, and it's in a protein coding region, if it sees ATG, that tells it, aha, this is the beginning of the code of the protein. And when it gets to one of the stop codes, it's, okay, this is the end of the code of the protein. So everything in the middle, it uses to specify amino acids to build the chain of the protein. So let's suppose then that we have a DNA chain and we're trying to build a protein. Well, what's the odds that we get the first amino acid right? So we would suppose this protein needed a certain amino acid, maybe proline or something. Well, suppose we needed C, C, and A. That's proline. So we needed that combination to get the first amino acid right. Well, since there's what's the odds that that would be the right code by chance? Well, since there's 20 amino acids, the chances are roughly 1 in 20 that we'd have the right code for the first amino acid. What are the chances we'd have the right code for the next 3 bases down the road for the next amino acid? Well, again, there's 20 amino acids, so the chances that we have the right code by chance for the next amino acid would be 1 in 20. And likewise, what are the chances we'd have the right combination of bases for the next amino acid? Again, it's 1 in 20 and so forth. So this is like our word information, remember? It's just to get probabilities, we multiply together the probability of each unit or each letter. You can think of amino acids as letters in an alphabet. So suppose we let the letter A represent an amino acid. what's the chance we get the first amino acid correct just by random chance? If we're just setting up DNA, picking up a random basis to set it up. Well, there's 20 amino acids. The chances that you'll get the correct code for the first amino acid is 1 in 20. Likewise, what are the chances you'll get the second amino acid correct if you're just ordering DNA randomly? Well, it's 1 in 20. Likewise, the probability you'll get any amino acid corrected in the correct order in DNA is 1 in 20. So what's the chances of spelling a protein by chance? Remember, in the word information, we had 26 letters, so it was 26 times itself 11 times. Well, in a protein, in a typical protein, there's like 300 amino acids. So we'd have 1 in 20 times itself 300 times to see the probability of getting a typical protein, the DNA for a typical protein by accident. So of these 300 amino acids, there's been a published paper done with actual living organisms, swapping out different amino acids in what's called a conservative way, to see how that reduces the viability of enzymes, which are a particular kind of protein. And it's been found that at least 80% of the amino acids must be correct for the protein to function. And actually, this was a very conservative study. They purposely avoided what we call active sites in proteins, and they purposely avoided interior sites in proteins. When proteins fold, it's thought that the interior sites, if you change the amino acids in one of those, you're more likely to damage the protein than if you change an exterior. So this study was very conservative. It didn't change any active sites. It didn't change any interior amino acids in the protein. And it only used what are called conservative substitutions. It only used, if it traded out an amino acid and substituted another, it only used amino acids that are conserved. And I won't go into that too much, but just it only made substitutions that are most likely to be conceived or compatible. And based on this study, which is actually done with living organisms and is published in a peer-reviewed journal of molecular biology, it is found that at least 80% of the amino acids must be correct for the proteins, typical proteins like enzymes, to function correctly. The number is probably much higher. Now, before this, it's been falsely assumed that evolution is true, and they looked at certain databases of proteins, tried to group them into families, and based on what they saw in certain families, they made certain assumptions about what could be substituted for what. But they didn't try it out with living organisms. This study is different. It actually does real science. It doesn't start with an assumption. It does real science, collects real data, and that's where they get this 80% number. The reference for this is the Journal of Molecular Biology. It's published in the year 2000, and it's an article. It's called "Conservative Amino Acid Changes on Enzyme Exteriors." By Douglas D. Axe, A-X-E. You can look that up if you want. So what's the chance that we get a protein, the right amino acids in the right order for a typical protein? Well, if you need 240 amino acids in the right positions, in the right order, odds are 1 in 20 times itself 240 times. That is a number that is beyond our comprehension. It's like a 1 with 312 zeros after it. Just by way of comparison, there's about 10 to the 22, that's a 1 with 22 zeros after it, of sand grains on all shores. There's about a 1 with 26 zeros after it of water drops in all oceans. There's about a 1 with 80 zeros after it of atoms in the visible universe. It requires a 1 with 312 zeros after it to spell a typical protein by chance. But it's worse than that, because with few exceptions, the machinery of the cell consists of assemblies of proteins, or less frequently individual proteins. So one protein will do you no good at all. You understand? And even if you had the DNA for a completed organism, the DNA would do you no good at all. You need not only the perfect DNA for all the proteins required for life and replication and digesting things, restricting what comes into the cell and what goes out of the cell for the waste mechanisms, ribosomes, you name it. Even if you had the right DNA, it would do you no good unless you had a whole cell, a whole factory to replicate the DNA. You understand? It's an intractable, impossible idea that life could have started by chance. Now, we want to take it a step further. We want to calculate not just the possibility or probability of getting the right code for a single protein by chance, but we want to calculate the probability or possibility of that all the matter in the visible universe, let's start on Earth. We'll start on Earth. Could, over time, come up with the right combination of DNA for a single typical protein. So we can't replicate evolution. Supposedly evolution took place over millions or billions of years. We can't do a science experiment, but we can do a thought experiment. When Albert Einstein did not have the scientific equipment to do certain experiments, he did thought experiments to rule things in and rule things out. So we're going to do a thought experiment. It's called a best case analysis. A best case analysis is if we know something doesn't work in the best case, we'll know it won't work in any case. So how do we do that? What are the odds of creating life on Earth by chance? Best case, or a single protein on Earth by chance. Well, we're going to assume every place on Earth is perfect for life. That's the best case, right? We're going to assume the Earth is 100 billion years old. Nobody thinks it's that old, but we're going for the best case, making assumptions favorable to life forming by chance. And we're going to assume the greatest possible number of atoms in the Earth. And then we're going to assume that every atom on the Earth is a computer programmed in the language of DNA, guessing combinations of DNA at a rate of 1 billion combinations per second. Now, that's just an absurd assumption, but we are assuming that every atom on Earth is a computer, and it's programmed in the language of DNA, guessing at a rate of 1 billion times per second. So let's take a look at our first assumption. First assumption is all of life is perfect for life. All of Earth is perfect for life. Well, actually, the center of the Earth is a hot rock core, or perhaps iron, but you can't have life there. It's only in a very thin layer on the surface that you can have life. So that is a very generous assumption on our part, that all of Earth is perfect for life. And we assumed a huge age for the Earth. We assumed 100 billion years, but all estimates of the Earth's age are less than 5% of that. And we assumed that each of these black circles is supposed to be an atom in the Earth. We assumed that every atom in the Earth is a computer. And we assumed that it's a computer programmed in the language of DNA. That's another absurd assumption. But see, these are all favorable to evolution. So what then would be the chances that we get the right combination? How many combinations would we get if every atom on Earth was calculating at 1 billion combinations per second for 100 billion years? How many combinations would we have in the best case? Well, we get a 1 with 87 zeros after it. That's a big number. 1 But is it big enough? No, we've already determined and calculated that you need the probability of getting the right combination of DNA for a single one typical protein we're talking about here is a 1 with 312 zeros after it. So could we get life on Earth by chance? Well, that's just impossible. That is impossible. Not a single protein could we get by chance. So once you have simple life, it's also impossible to create the information for new kinds of life by mutations plus natural selection. And we show that in another video. So there's two problems. One is to get life started. That's an insurmountable problem. Even the staunch atheist evolutionists say we don't know how it happened. The reason they say they don't know is because it's not scientifically possible. Chemistry prevents it. Information science prevents it. So even people like Richard Dawkins admit they don't know how. And we're going to deal with that in a later video. We're going to actually show you an interview with Richard Dawkins, one of the most famous evolutionists alive today. And also we're going to show you some quotes from the man, Sir Francis Crick, who was a co-discoverer of how information is stored in DNA. He's if anybody ought to know about it, it's him. And I think you'll find it very interesting how they respond to this problem. So since the creation of the world, God's invisible attributes are clearly seen, being understood by the things that are made, even his eternal power. The Bible tells us that if we examine nature, the hand of God is clearly evident. I'll tell you, it's been such an encouragement to me as I have studied this question of evolution versus creation, how overpowering the scientific proofs are to show that life did not happen by natural means. It's been really amazing. And also that life could not evolve into new kinds of creatures by natural means. The evidence is overpowering. All this points to our great Creator God. Jesus Christ is his name. I hope you get an interest in the Bible, because the Bible is the one book that gives you an accurate history of how life came to be supernaturally, and also about your eternal destination. You will meet your Creator as you read the Bible and study the pages of it. A great place to start would be the book of Mark in the New Testament. Go to BibleGateway.com. Start reading the book of Mark. You'll learn about Jesus' life and teaching, his death for your sins, his resurrection from the dead, and the way to obtain forgiveness of your sins and eternal life. So the Bible tells us without him, without God, nothing was made that was made. You can give your own presentations. If you like a PowerPoint of the presentation you just saw, you can go to the website of educate7.com. Look for the Are You an Accident of Nature series. You can watch this video free. You can download the PowerPoints free, and I encourage you to do that. Share them at your university or your school, with your friends, your family, a church group. You're free to use these PowerPoints. Our next presentation is Are You an Accident of Nature, Part 5. It's on leading evolutionists and their speculations when faced with this dramatic information that disproves evolution. They say, maybe aliens created life. I kid you not. You'll see an actual video of that, and I'll show you some other published information on that. You cannot form life anywhere in any universe. These same chemical problems, the same information science problems, the same entropy problems apply anywhere in any universe. So this is not a legitimate way to believe. But I hope you'll be led to check that out. And thank you for watching.